Welcome to the chapter on biochemical engineering. This field combines principles from biology, chemistry, and engineering to develop processes and products that can improve human health, the environment, and industry. In this chapter, we will cover enzyme kinetics, bioreactor design, and downstream processing. Enzyme kinetics is the study of the rates of chemical reactions that are catalyzed by enzymes. Understanding enzyme kinetics is crucial for designing and optimizing biochemical processes. One of the most important models in enzyme kinetics is the Michaelis-Menten equation, which describes how the rate of the reaction depends on the concentration of the substrate. The equation is given by V equals V max times the substrate concentration divided by Km plus the substrate concentration. In the Michaelis-Menten equation, V represents the initial reaction rate, V max is the maximum reaction rate, S is the substrate concentration, and Km is the Michaelis constant, which is the substrate concentration at which the reaction rate is half of V max. Consider an enzyme-catalyzed reaction, where the maximum reaction rate, V max, is 100 micromoles per minute, and the Michaelis constant, Km, is 5 millimolar. If the substrate concentration, S, is 10 millimolar, the initial reaction rate, V, can be calculated as follows. V equals 100 micromoles per minute times 10 millimolar divided by 5 millimolar plus 10 millimolar, which equals 1,000 micromoles per minute divided by 15 millimolar, resulting in 66.67 micromoles per minute. A bioreactor is a vessel or container in which biological reactions are carried out. The design of bioreactors is crucial for the efficient production of biological products, such as pharmaceuticals, biofuels, and food products. There are different types of bioreactors, including the stirred tank bioreactor and the airlift bioreactor. The stirred tank bioreactor is the most common type of bioreactor. It is equipped with an agitator to mix the contents and maintain uniform conditions. It is used for both aerobic and anaerobic processes. The airlift bioreactor utilizes a stream of air to circulate the medium and maintain mixing. It typically has lower shear stress compared to the stirred tank bioreactor, making it suitable for shear-sensitive cells. Key parameters in bioreactor design include the volumetric oxygen transfer coefficient, KLA, and the residence time, tau. The volumetric oxygen transfer coefficient measures the efficiency of oxygen transfer from the gas phase to the liquid phase. This parameter is critical for aerobic processes where oxygen is a limiting factor. Residence time is the average time a substrate molecule spends in the bioreactor. It is given by the formula tau equals the reactor volume divided by the volumetric flow rate. Consider a stirred tank bioreactor with a volume of 1,000 liters and a volumetric flow rate of 100 liters per hour. The residence time, tau, can be calculated as follows. Tau equals 1,000 liters divided by 100 liters per hour, which equals 10 hours. Downstream processing refers to the purification and recovery of biosynthetic products, particularly pharmaceuticals, from natural sources such as animal or plant tissue or fermentation broth. Key steps include cell disruption, separation, and purification. The key steps in downstream processing are cell disruption, separation, and purification. Consider a fermentation process producing an antibiotic. The downstream processing steps might include cell disruption using a high-pressure homogenizer to break open bacterial cells, separation by centrifuging the homogenate to remove cell debris, and purification using ion exchange chromatography to purify the antibiotic. This concludes our chapter on biochemical engineering. By understanding and applying these principles, biochemical engineers can design efficient and effective processes for producing a wide range of biological products. Thank you for watching.